Homer Smith was a visionary.
He was curious. He was innovative.
He was foundational to modern nephrology.
The Homer Smith Award of the American Society of Nephrology was the first award given, and it reflects Homer Smith's dominant role in understanding the physiology of the kidney.
Smith led the Department of Physiology for decades at NYU, and it was Smith, probably more than any other person, who defined the modern concepts of glomerular filtration, tubular excretion, and also secretion.
Homer Smith truly inspired generations of physicians, including my own.
He brought to nephrology principles upon which our field is based.
All of these basic things inform our care of patients.
He used comparative biology in order to advance our understanding of more complex systems, the human kidney.
He brought to NYU techniques and information that he had gathered by studying fish. Many of those experiments were carried out in Mount Desert Island.
The Mount Desert Island Biological Lab played an extraordinary central role. Smith realized that marine organisms gave him powerful tools to study the evolution of the kidney.
He was into philosophy, music, science in the broadest sense, religion.
To read him, it sort of opens your mind to other areas and it fires the imagination.
When he said something, it turned out to be correct because he was very precise in what he did. And that's a lesson for all of us.
He really set the stage for all future researchers and how to move the field forward.
The American Society of Nephrology presents the 2024 Homer W. Smith Award to Dr. Joseph Bonventre for his seminal research contributions and leadership in science and nephrology. He is the Samuel A. Levine Distinguished Professor at Harvard Medical School and Constantine L. Hampers Distinguished Chair of Renal Medicine, Chief of the Division of Renal Medicine, and Founding Chief of the Engineering and Medicine Division at Brigham and Women's Hospital. He was Harvard Director of the Harvard-MIT Division of Health Sciences and Technology. Born in Brooklyn, Dr. Bonventre earned a B.S. with distinction in engineering physics at Cornell University, his M.D. at Harvard and MIT, and a Ph.D. in biophysics with Claude Lechen as his mentor. He completed postdoctoral training at Harvard and Massachusetts General Hospital. Dr. Bonventre's research focuses on the study of kidney injury and repair and signaling transduction, with a special emphasis on the role of inflammation, biomarkers, and stem cells. He has authored over 600 publications and 3 books. His work has been cited more than 96,000 times. Among his early contributions were pioneering studies regarding the role of calcium in mesangial cells and acute kidney injury, AKI, as well as the cell signaling role of phospholipase A2 and early studies of gene expression with AKI. His laboratory identified the role of sex and susceptibility to AKI, the importance of inflammation in AKI, the role of senescence, and the cells involved in adaptive and maladaptive repair after kidney injury. Dr. Bonventre and his collaborators influenced the definition of acute kidney injury. He identified a key general transcriptional inhibitory domain that subsequently enabled CRISPR-I technology. He cloned and characterized kidney injury molecule 1, KIM1, as a sensitive diagnostic and prognostic biomarker of kidney injury and progression of chronic kidney disease and kidney cancer. He and his lab have extended his kidney repair and stem cell work to create human kidney tissue models of disease. His protocols to generate kidney organoids have led to a revolution in kidney research using human tissue. Dr. Bonventre's work has been recognized with numerous honors, including the International Society of Nephrology Bywaters Award, the Osler Medal of the Royal College, and 2 Merit Awards from the NIH-NIDDK. He has been elected to the American Society for Clinical Investigation, the Association of American Physicians, the American Institute for Medical and Biological Engineering, and Fellow of the American Association for the Advancement of Science. A past president of ASN, He is currently on the editorial board of Jason, See Jason, and Kidney 360, and is editor-in-chief of Seminars in Nephrology. He has played a number of leadership roles in ASN initiatives over the years. He has been awarded honorary doctorate degrees from the Norwegian University of Science and Technology and Mount St. Mary's College. Dr. Bonventre and his wife, Dr. Christina Cannon Bonventre, have 2 children, Andrew and Joanna, son-in-law Brian, and 3 grandchildren, Daniel, Alex, and Taylor. The family, including Joe's parents, Fanny and Vincent, along with his friends, mentors, trainees, and colleagues worldwide, have long supported Dr. Joseph Bonventre in his various roles as scientist, educator, clinician, and as a foremost contributor and leader in nephrology.
Please join me in welcoming the Homer Smith Award recipient for this year, Dr. Joseph Bonventre, who will present on kidney injury and repair.
Well, that's sort of amazing. I'm really humbled and greatly honored. I want to thank the ASN. I want to thank Dr. Cruz, Dr. Josephson, and all my colleagues over the years and trainees. This is an incredible award, the most prestigious I've certainly ever received, and suspect the most prestigious I ever will receive. So now I'd like to start my presentation. Let's see, okay, and start with these disclosures. And then I want to say that, clearly this award really belongs to all my former fantastic trainees and colleagues and collaborators. On the next 3 slides, I've illustrated approximately 1/2 of these trainees. I think you'll recognize many of them because many of them have gone on to great things in medicine, in nephrology, and in other fields. I also want to recognize my mentors. As you heard, I was an undergraduate at Cornell in engineering physics, and then my life was transformed by an experience in the summer program at UCLA and Caltech and the Jet Propulsion Laboratory on space biology. And I came back from that as a rising senior in college and decided that I wanted to do something related to biology and medicine. I then applied for graduate school and was accepted by Arthur Solomon at Harvard in the Harvard Biophysics program without any biology course on my application. So I came to Harvard as a graduate student initially, and then looked around and looked for like-minded people. There were at the time two biomedical engineers at Harvard College, and one of them was Tom McMahon, who then directed me toward Ryan Bucas and Cliff Barger in the physiology department at Harvard Medical School, who introduced me to the kidney. I then, as a graduate student, evolved into someone who really wanted to do medicine also. And at that point, Irving London accepted me into this new program, which is the Harvard-MIT Division of Health Sciences and Technology, which I subsequently had a leadership position in. And my PhD advisor was Claude Lechen, as you've heard. I finished my PhD on renal concentrating mechanisms and then went to the MGH for residency, and where my research and overall mentor was Alex Leaf and clinical mentor Pete Coggins. Stayed at the MGH for 25 years or so, and then Victor Zhao recruited me to the Brigham to replace this man, Barry Brenner, whose name has already come up. And Barry, of course, is someone who can't be replaced, who could never be replaced. And unfortunately, Barry had passed away this summer. but he contributed such an enormous amount and was an incredible role model for what one person can do in an entire field. I also want to thank my colleagues at the Brigham. This is one picture of the large division that I've been having the privilege of working with over the years. Many of these people who are still there or who have left and gone on for leadership positions at other institutions throughout the US and the world. I also want to recognize my other division of engineering and medicine, where Victor Zhao was at least tolerant of my idea about establishing this. This is, I think, the only such division in a department of medicine in the country. And right now it has about 140 faculty, trainees, and associates. So now let me turn to the science, and there's been a lot that I've been involved with, and so I'm going to actually present a sampling, truly a sampling, years worth of work summarized in one slide in many cases. And I'm going to mention some of the fellows. I'm going to call them out, or at least by having a picture on the slide. But of course, in anything I mention, there are many, many trainees and colleagues who have been involved. So my career has basically been associated with injury and repair in kidney, and driven by these four questions. One, how do we best identify injury to the kidney? What are the primary pathophysiological contributors to the injury? How does the kidney repair itself in an adaptive way? And why does the repair process become maladaptive? Can we develop ways to restore, create human kidney tissue and better model human kidney diseases in our constant quest to find new therapeutics for our patients? So I'll start actually with a collaborative study. Again, collaborators have played an enormous role in my career. And this is with Glenn Chertow, and actually was initiated in the airport, an airport waiting room, when the two of us were coming back from a meeting. And this study essentially demonstrated the importance of small changes in serum creatinine, 0.3 milligrams per deciliter. With an odds ratio for death, just looking across the Brigham database over a year, odds ratio for death of 4.1. And this study helped to influence the change in the way we thought about acute renal failure at the time, and led to the change. You've heard about ADKI and the role of Claudia Ronco and others in ADKI. One of the groups that came together to define kidney injury, acute kidney injury, and then subsequently the KDIGO definition. Both of these definitions took into account these small changes in creatinine so that you didn't have to get failure to really have a significant injury to the kidney that was meaningful for patient survival. But of course, creatinine, we all know, is not a great marker for acute events in particular, as reflected on the right-hand side of this slide by the relationship between creatinine and glomerular filtration rate, one marker of function of the kidney. So now I'll move into other biomarkers and other ways to look at injury, and particularly focus on kidney injury molecule 1, which Hari Ikamura in the lab, he's still in the lab with me as a faculty member, but as a fellow, he, working with Michelle Sanicola and Veronique Bailey, collaborators, was doing some studies looking for genes that could be modulated in expression with ischemia. These studies, actually, I personally did these animal studies in the rat. And so we prepared messenger RNA from post-ischemic rat kidney and contralateral kidney that was normal or closer to normal. And then at the time, this was a time of when mRNA chips were popular. That was the leading technology. But we didn't jump on that bandwagon at the time. We used a completely unbiased PCR-based method. I won't go through the details, but it was an iterative method where you amplified the genes that were upregulated differentially between two populations. And with that, we discovered a molecule which we called kidney injury molecule 1. It's a single transmembrane domain molecule, heavily glycosylated and cleaved by metalloproteases, and appears in the human urine. And that was one of the driving forces for us to continue the work, because early on, we knew two things that we found were particularly interesting. First of all, it was in the proximal tubule. And second of all, it was clearly relevant for humans. It was also very interesting to us in that it was primarily a kidney-specific protein. And then Vishal Vaja, as a fellow, showed in the rat that the levels of KIM1 in response to cisplatin, the levels increased in a quantitative way in the urine well before there was any change in blood urea nitrogen or creatinine. So there's 1 to 2 days after cisplatin, there were marked increases. The baseline levels were extremely low. So marked increases in the levels of KIM1 when creatinine and BUN had not changed. And fast forward, we've done a number of studies in humans and tissue, but this is a more recent one with and as a collaboration with Sandro Sandagati and Jia-Young Chen at Harvard. And this demonstrates the utility of this molecule at a time, this human biopsy, at a time when the histology was relatively mild. This was a nephrotic syndrome patient who had minimal change disease. Minimal change, hence the relatively mild in terms of abnormalities, mild tubular dilatation, focal brush border loss, isolated mitotic cells, all characteristics of kidney injury, but relatively mild. And yet you look and see the very dramatic pink color of practically all of the proximal tubules. And then subsequently working with Predictive Safety Testing Consortium, which was a consortium primarily of pharmaceutical company representatives looking for new markers to help in drug development. We looked at 10 different nephrotoxicity models established by different companies and looked at a number, approximately 10, of biomarkers. And here is a summary at the top of all of the data as a function of the area underneath the curve, of the receiver-operator curve, and with varying amounts of histopathology. So 0 to 1 meaning that there's very little histopathology in the rat kidney, and yet KIM1 performed incredibly well, the ideal biomarker being one that has a or has an area underneath the curve of 1 in the way of representing this. And certainly when there was little injury to the kidney, there were BUN or N-acetylglucosaminidase, NAG, or the serum creatinine did not really perform very well. And so this was published here, and then later that year, a European consortium came to the same choice of showing that KIM1, again, completely independently, turned out to be a very good marker for injury in the rat here. So in 2008, KIM1 was qualified by the FDA and the EMA in Europe, and also the Japanese Regulatory Society, as a preclinical safety marker for assessing kidney toxicity of experimental drugs. And actually, a number of years later, the FDA published a paper looking at the regulatory applications after this qualification, and you could see that when, at the time, the molecule was qualified, subsequently the number of regulatory applications that reference KIM-1 as a drug development tool increased substantially. In 2018, more recently, KIM-1 was qualified by the FDA as part of a composite to use in phase 1 clinical trials to evaluate nephrotoxicity. This was based on studies of gentamicin and cisplatin in humans. Okay, so that's urine. We're talking about urine KIM-1. Venkata Sabasetti, Sabi, when he was a fellow in the lab, looked to plasma KIM-1. So this was now around 2014, and found that plasma KIM-1 increased with ischemic injury to the kidney. Even periods of 10 minutes of ischemia or 20 minutes of ischemia, which had essentially no effect on creatinine 24 hours later, produced significant increases in plasma KIM-1, in addition to what was expected already at that time, the absolute urinary KIM-1 levels or the normalized urinary KIM-1 levels, normalized to creatinine. And in collaboration with Natalia Nowak and Andrei Kryluski, we also showed in that paper that plasma KIM-1 was a good predictor of bad outcomes, essentially, in a population of patients with type 1 diabetes. So if the plasma KIM-1 was above a certain level, that patient was much more likely to develop either end-stage renal disease. And so that led to a number of other studies. This was one we were not involved with, but this was a European study with 2 diabetes cohorts where they looked at 297 blood markers and compared those markers as prognostic of the decline in renal function. And this was adjusted for many clinical covariates. And of the 297, 2 sort of stood out, one being KIM-1 and one being member of the TNF receptor family. And here's a number of papers, or not a complete list, but a number of papers that involve or show that plasma KIM-1 is a prognostic biomarker for progressive kidney injury or cardiovascular disease or mortality. The studies in yellow were studies from the CKD biomarker consortium that I was privileged to be associated with. Okay, and another aspect that's been interesting and timely right now is that we discovered many years ago, we expected this, actually, because we knew a lot about the molecule, that it's markedly upregulated in renal cell carcinoma and in lymph node metastases. And fast forward to a paper that we recently published in collaboration with Vincent Zhu, junior faculty member at the Dana-Farber rather, showing that with increasing levels of plasma KIM-1, you could predict a bad outcome in renal cell carcinoma. And this has also gotten a lot of press at the ASCO meetings. And here I've just quoted the non-invasive biomarker KIM-1 is front and center in renal cell carcinoma investigations. And Tony Kaluri, who's the leader in this field, sent out an survey of what's the most important biomarker in renal cell cancer. And it turned out of these 4 choices, within an hour, he got 200 replies on KIM-1. Okay, now let me move on from KIM-1 to primary pathophysiological contributors to injury. So we started out, as you heard, really studying intracellular calcium at a time when this was actually the first representation of using fluorescent dyes, Quin2 and Quin2 and Fura2, to measure intracellular calcium both in epithelial cells of the kidney and in mesangial cells. And at the time, we also were studying phospholipase A2 because we thought that that was a primary contributor to mesangial cell injury. And this was a time when, if you did a knockout, you could get published in Nature. So you see how old these data are. Okay, and then it was mentioned on in the video that we also early on looked at the role of sex and showed that males were much more susceptible than females, and that we could introduce a male phenotype to intact female mice by giving them testosterone. And then we did orchiectomy, not on this slide, in males and converted them into a female protected phenotype. And more recently, Morgan Grams and her group looked at over 1.2 million participants in meta-analysis, and also demonstrated in humans that, again, the mouse studies were predictive of the human outcome, that females are less susceptible than males to acute development of acute kidney injury. So at the same time then that all of this other work was going on, again, we were very interested in molecular characteristics. What made the post-ischemic kidney more susceptible to injury, or what are the characteristics? What genes were upregulated? And here was Ralph Wirtschgall cloned a gene that we called KID1, wasn't known at the time, so we got to name it, for kidney injury and developmentally regulated gene 1. And that gene had a number of zinc finger motifs, and we found that it was a transcriptional inhibitor. We then mapped the region of the gene. These are the representation of the protein in different domains. We mapped the region of the gene that was responsible for the transcriptional inhibition using a CAT readout here, where you attach the part of the gene protein to to GAL4, and that binds to binding sites on the reporter construct. And then we found proteins that interacted with this domain, which was responsible for the inhibition. So looking at these here, the inhibition of transcription. And it turned out, fast forward 15 years later, that others have used this CRAB domain as a basic principle for CRISPR-I technology of suppressing transcription of particular proteins in general. We spent a number of years on inflammation, and this is a reflection of all of those years, both looking at the endothelium and looking at the role in the tubule. And here, back in 2004, we concluded that ischemic acute kidney injury, or acute renal failure at the time, was an inflammatory disease. And this has been confirmed by many others, including in a paper in Nature by the KPMP consortium. Okay, how does the kidney repair itself in an adaptive way, and why does a repair become maladaptive? Early on, again, Ralph here, showing that as part of the repair, there was a proliferative response. And in addition to proliferative response as measured by multiple markers, there was also a transition. The epithelium took on characteristics of the mesenchyme, and subsequently, in this case, vimentin. And so then we also, early on, did a number of studies on preconditioning. And here, this phenomena of preconditioning was really profound, in the sense that if you made the mouse ischemic, if you made the mouse kidney ischemic, the creatinine went up, came back down. This was bilateral ischemia. If you then came along 8 days later and took the same mouse and you increased the period of ischemia, there was no change in serum creatinine. So there's clearly a lot going on, with even persistent up to 8 days, that was changing the response of the kidney. And so that was what we were looking for, with mechanisms or processes that we could then recapitulate using pharmacological tools. But the other thing that we found at the time was at a time when basically people thought that after ischemia, the kidney completely repaired, but 12 weeks later, even though creatinine had been way down pretty much by 5 days after the ischemia, there was persistent activation of myofibroblasts. And so this was a very early indication that acute kidney injury could lead to more chronic changes in the kidney. And then Ben Humphreys and Jeremy Duffield in the lab, basically in collaboration with Andy McMahon, worked on this whole issue of what cells are involved in the repair process. So we know that there's a lot of proliferation, there's a dedifferentiation in the epithelium, loss of polarity. Did the cells that replenish the lost cells come from the bone marrow? Did they come from interstitial stem cells, or did they come from stem cells in the epithelium itself? And we actually found that they didn't come from any of those sources, but they came actually from proliferation of the surviving epithelial cells left after the cell death. And then again, looking at different mechanisms of injury here, when Li Yang was in the lab with Guillaume Queneau and Seiji Akishi and Craig Brooks, developed this whole notion of senescence, that in fact, what happened with injury was that the cells went into a cell cycle arrest period, and they then produced molecules that were profibrotic. And this shows with moderate IRI, there's a return of different phases of the cell cycle that the cells respond, that are entering the cell cycle, whereas with aristolochic acid or unilateral ischemia reperfusion injury, where there was much more fibrosis, that the cells did not respond, there was not a return, there was a persistent senescence. And this has led to a number of studies by many groups looking at senescence as a major contributor to the abnormal repair that goes on after injury, and ultimately resulting in progressive fibrosis and chronic kidney disease. Okay, let me just say that KIM-1 also plays a role here. KIM-1 under normal circumstances is a phosphatidylserine receptor. It's an anti-inflammatory response of the proximal tubule to slow down inflammation, as reflected by this cartoon where the apoptotic bodies are taken up because they're coated with phosphatidylserine, and this ultimately results in antigen presentation and upregulation of Tregs, downregulation of inflammatory immune cells. But more prolonged levels of KIM-1 expression in studies honchoed by Yutaro Mori and Hari, this becomes maladaptive by adding to lipotoxicity, mitochondrial stress, DNA damage, cell cycle arrest, and profibrotic mediators. Okay, and then the last, I'm a little over, so there's a few more slides, please. But I want to say that essentially, how do we create, restore kidney tissue? You're going to hear a lot of this in Melissa Little's talk that's coming up. But I want to just say that in studies initiated by Albert Lamb and then greatly expanded and brought to conclusion by Benjamin Friedman and Ryuji Morizani, this is an example of the generation of kidney tissue with markers of the glomerulus, the proximal tubule, and the distal nephron in studies. Ryuji was the primary author here, first author. And at close-up, you can see the generation of what looks like Bowman's capsule around a podocalyxin positive tuft leading into a proximal tubule. And here, a proximal part with a distal part, the V-cadherin, with TAM-horsfol protein in the middle. And EM, there's very interesting structures that are formed, looking like podocyte bodies, as well as basically villi and intercellular junctions. Bino Friedman developed at the simultaneously published the same year another protocol, and he basically then has developed this, then used CRISPR technology to knock down PKD1 or PKD2 and establish this as a model for cyst formation, and he's gone on to really develop this in an incredible way, looking for new therapeutics. And then finally, in terms of from a science point of view, this was Kyle McCracken and Min Shi in the lab, who developed a technology to develop collecting duct organoids and ureteric bud organoids. And you're going to hear, I think, maybe a little bit of this from Melissa also. But this technology results in, when examined by single cell analysis in a population of cells that are primarily cells of the collecting duct to principal cells. Okay, I want to thank, again, the mentors, my trainees, my grant reviewers, who have been, when they were kind, they were, but when they weren't so kind, they also motivated me to prove them wrong. And so I thank them. And the funding agencies, in particular, the NIDDK, which has supported me over the years, and actually a lot of what I presented was based on 1R01, which is in its 39th year. And these are some of the collaborators, only ones from the 3 consortia. Again, there are many, many others. I want to thank again ASN. This is a year from my council group when I was president. I want to thank Todd. And when I was president, that was the year that we modified this celebration from Renal Week to calling it Kidney Week, which I'm also proud of. My family, you've already seen pictures of my daughter, Joanna, my son, Daniel. Yeah, Andrew, you get that confused a lot. And my 3 great grandsons, Daniel, Alex, and Taylor. Alex and Taylor are here. Joanna's wife, Brian, my mom, who's celebrating her 99th and a half birthday, and my dad, who's unfortunately not with us now. And of course, my wife, who's been with me the whole time, and thank you. We've just celebrated 2 weeks ago our 50th wedding anniversary. So thank you all. Sorry.